Welcome to our ultimate webinar. I've been waiting a while to say that. Now, we have a slight technical problem, so we won't be able to see James today unless miracles happen during the course of the presentation. So, brief overview. The slides for this presentation are already uploaded. They're linked through the event link on the front of the website. The document has just been updated, so if you went to look at that link half an hour or so ago, it would have been a relatively sparsely populated document. It is now full of interesting words, so I encourage you to go back to that. This session is being recorded. Everything you say and type is going to be put out onto the internet, and I think that is about that. So I'm going to fade myself into the background and pass over to James.
Okay, well, what I want to talk about is some what I think are relevant and pressing questions on the topic of the origin and early evolution of photosynthesis. You can see the first slide there, introductory slide. Just some very broad questions. How soon after, what we're trying to address here is how soon after the origin of life did photosynthesis arise? Did simpler forms of light-driven energy transduction precede anoxygenic photosynthesis? We are pretty sure anoxygenic photosynthesis preceded oxygenic photosynthesis. When did organisms evolve oxygenic photosynthesis? And of course, we have a fair bit of geological constraint on that topic, and I'll mention some of that. Now, the title of this is Origin and Early Evolution of Photosynthesis, and what I mean by early here is I'm basically just going up to the Great Oxidation Event. So something on the order of 2.4 billion years or so in age. I'm not really focused on photosynthetic eukaryotes and endosymbiosis and all of that, which is all very interesting and very complicated in some ways. At least the eukaryotes are very complicated. So I'm just not really prepared to talk about that. There is, of course, the interesting aspect of oxygen rise in the Neoproterozoic, which almost certainly involves photosynthetic eukaryotes. So let's going to skip over that. Okay, so moving on to the next slide. Just a summary of the net reactions of photosynthesis. The first reaction you see there on the top. Now, I suppose, I don't know, there's a pointer. Okay, there we go. Yes, there it is. Okay, so you should be able to see that green arrow if you're on Adobe Connect. And you see just the sort of generic net reaction for photosynthesis, CO2 plus 2H2A, where A can be sulfur or oxygen or nothing, must be hydrogen, plus photons yields a reduced carbon, in this case written in the form of what looks like formaldehyde, but it's really glucose and things of that sort, plus water, plus some oxidized component in the original reductant. So that's just the generic reaction for photosynthesis. Oxygenic photosynthesis, of course, the H2A would be H2O, we'd water, and we'd be liberating oxygen, which is obviously the source of it in our atmosphere. Writing this net reaction out in a bit more detail, you see here the more detailed oxygenic form of it, showing that you're actually making glucose. Therefore, of course, you need more of everything. And it just gives a bit more detail and traces the oxygen. So the oxygen in the CO2 goes into the glucose. The oxygen from the water goes into oxygen, molecular oxygen in the atmosphere. Okay, anoxygenic photosynthesis is also very important, and as I already mentioned, probably preceded oxygenic photosynthesis, really Earth, and there are a number of reductants possible there, including sulfides, hydrogen, I should use the pointer, using the wrong pointer, sulfides, hydrogen, and ferrous iron, all of which, especially the ferrous iron and sulfides, very likely to have been abundant on the early Archean Earth. And so these are all relevant processes as well. Okay, going on to carbon isotopes. So this is what I'm going to do is just give a brief introduction to what geological, and this is very brief, but a brief introduction to what geological constraints we have on photosynthesis and its occurrence. And some of the most important of those are, of course, the carbon isotope record in sedimentary rocks. And by that I mean comparison of the carbon isotopes in carbonates versus organics in the same rocks. So what you see here is I just swiped this from the web. I didn't really have a handy plot for this. And what you see here is delta C13 of the organics relative to, this should be PDB, PD belemnite carbonate, and you see that everything is negative. So all these organics are negative. They're negative in the range of kind of mostly in the range of minus 20 to minus 40 per mil relative to carbonate. And some of them going even lower, minus 60 or so per mil. And this is all interpreted as evidence for carbon fixation. And that carbon fixation could be via photosynthesis, or it could be by some other form of autotrophy. When we talk about very old carbon isotopes, we don't really know what it is. We know better when we go into the mid-Proterozoic that it's really photosynthetic, and it certainly is in the Phanerozoic. Oxygenic photosynthesis is creating this distribution of carbon isotopes. These oldest samples right here, which should be from the Achelia rocks, or at least some of them are from Achelia, maybe some from Isra. These are likely, these are quite metamorphosed rocks, and so it's not entirely clear what has happened there. It is possible that this is evidence for oxygenic photosynthesis that long ago. We don't really know exactly how to interpret these. There have been some other suggestions for how one could do this abiotically, in fact, but that's not clear. Okay, but the main point is that there's a consistent record over quite essentially all of Earth history that we have in carbon isotopes of some kind of carbon fixation and quite possibly photosynthesis. Okay, these are figures from Bill Schaaf's review paper in 2011 looking at the sites at which microfossils are collected and the carbon isotopes in the organics associated with those rocks. In addition, I will show the actual microfossil data in a minute. But again, you see that in a number of these old sites from which microfossils are collected, there are also similar carbon isotope values, so on the order of minus 20, minus 30 per mil, relative to the carbonates, EB in this case written correctly, and same up here. So this is just a broader span of time, but again, you see the same general trend. So the interpretation generally is this is evidence for oxygenic, or at least photosynthesis. Need not be, this down here need not be oxygenic photosynthesis, but it certainly some kind of photosynthesis. But again, we really can't, just based on these carbon isotope values alone, rule out other forms of chemolitho autotrophy, things of that sort that can also produce reduce carbon with low C13. Okay, these are some of the microfossils. Again, just reviewing some of the kind of evidence, or what is presented as evidence for photosynthesis. These are from the apex chert, about 3.45 or so billion years in Western Australia. Various photomicrographs and analyses in Raman arguing, really, Schaaf is arguing here for the similarity of these organisms, what he thinks are microfossils, to cyanobacteria. And it's certainly a possibility. We don't know. I think it's entirely clear. Schaaf has definitely made the case that these are truly organic containing microfossils. There was quite a debate about that. Whether they are cyanobacteria or not is, of course, that is a much harder thing to determine. May not even really be determinable at this point, but morphologically they are similar. But as my more biologically inclined colleagues will point out there are many things that can look like this, and they need not be cyanobacteria. But the point is, these are in the rocks. They definitely contain organics, and it appears to be bioorganics. So it's very compelling evidence at 3.5 billion years for carbon fixers of some sort, and certainly photosynthesis, possibly photosynthesizers. Okay. So I don't know if you guys can all hear me. I assume you can't. Just jump in any time with questions. I'm totally receptive to questions here. These are some of the carbon isotopes. I think this is basically all of the carbon isotopes reported for various microfossils of different ages, so going from moderately recent stuff, Bitter Springs Formation, Australia, 850 million years, to Gunflint at 1.9 billion, all the way down to microfossils from 3.5 billion in the Dresser Formation, again, Australia, Western Australia. And same story. Basically, you see the same kind of minus 30-ish per mil, although these are getting quite light. Sorry, using the wrong pointer. These are getting quite light here. And with fairly large error bars, I don't really know what's been happening there. You do see a pretty consistent minus 30-ish per mil value for these actual kind of minus 30 to minus 40 per mil for these microfossils. The exception perhaps being the most recent, the Bitter Springs, which are more in the minus 20 to minus 30. Okay, but the point is these certainly look like evidence, again, for microfossils that are carbon fixers in some form.
Can I ask a question?
Yes, please.
This
say who you are.
This is Bob Blankenship.
Hi, Bob.
Hi. If you can go back to that. Yeah. The carbon fractionation in the various anoxygenic phototropes has been looked at, but not very extensively. Can you maybe remind us what sort of fractionation you get from things like green sulfur bacteria, purple bacteria, and so on, which have, at least the green sulfurs have a very different carbon fixation pathway?
Yeah, no, I do not have those numbers off the top of my head. My understanding is that they're really not tremendously different, but it's a reasonable question, and I certainly can add that. I haven't looked at that recently.
Yeah, that was my understanding too. But these that are down there about minus 40 are getting pretty light.
Yes, indeed.
I wasn't aware of those numbers.
No, I actually was not either until I pulled up this paper, and I have to say I'm frankly a little bit surprised about that. And I would say take this with a grain of salt. These are relatively recent results. I know these did not exist, as of 10 years ago or whatever. So they're relatively recent work, and we could talk more about that, but. I would definitely take them, as I said, with a grain of salt. I think there may be some other issues there.
There might be a place for arguing to acquire some additional data of this sort, which I think should be reasonably straightforward, and all the cultures of the various groups of anoxygenic bacteria are available in various laboratories. So it might be
it could be, but I would be just shocked if that has not all been done on extant organisms. I mean, that would be pretty surprising. But I really haven't looked. So yeah, it's certainly worth pointing out. I will make a quick note of that. Okay, let's see.
Hey, Jim?
Right here.
This is Kevin Redding.
Hi, Kim.
Hey, the only paper I know of was published in 1977. Bob Buchanan was on that, where they compared chlorobium thiosulfatophyllum with purples and Chlamydomonas for good measure. And I don't have the details, but chlorobium was different, as I recall,
okay,
than the other ones.
All right.
And interestingly, everything else uses rubisco, and chorobium does this reverse TCA cycle. So
chorobium tends to be enriched in carbon isotopes. This is Lindsay Hayes. Chorobium, because they use a reverse TCA cycle in the carbon fixation pathway, the carbons that they have tend to be enriched.
How enriched?
Enriched in what?
Enriched in carbon-13.
Okay.
But what
So you mean it's lighter?
Anywhere between, I don't know, 10 per mil or so?
So you mean minus 10 per mil, or 10
per mil
enriched?
over relative to other things that are using other carbon fixation pathways.
Gotcha. Right.
Yep.
That makes sense. Okay. So that is the error bar for many of these measurements on the microfossils, sort of 10 per mil. But that is a significant difference, for definitely something we should account for. Good point. Okay, let's move on. No more questions on carbon. Another constraint from the geological record is, of course, what record we have of oxygen in the atmosphere and the banded iron formations themselves, which is in this next figure. I lost my pointer. Hang on one sec. I don't see it. There it is. Okay, so the banded iron formations are here in red. Oxygen levels inferred are in blue. And so it's pretty clear that something was causing ferrous iron to crash out and deposit as magnetites and hematites in the Archean and early Proterozoic ocean. And exactly what the cause of that is still debated. And I mean, it is possible that it is, at least I think it's still considered possible, that it is simply the reaction of oxygen with ferrous iron, aqueous reaction of oxygen with ferrous iron, causing it to oxidizing that ferrous iron. Or it could be somewhat more interesting and involve anoxygenic organisms that use ferrous iron as a reductant and precipitate out FeOH3, and I think there is in fact iron isotope evidence. I did not get a chance to pull that up, but I will do that at some point. There is in fact I'm quite sure there's iron isotope evidence in favor of the latter, and there's even a third possibility, and that is that photolysis of ferrous iron complexes can also, you can photooxidize ferrous iron in surface water, although there have been some arguments, pretty substantial arguments against that. But the bottom line is that the BIFs it's a truly massive amount of iron, and one really does have to understand how it's formed, how that ferrous iron was oxidized. And it is either evidence for very early oxygenic photosynthesis, or it is evidence for the ferrous iron, the anoxygenic production by ferrous iron reduced oxidized. So it's an interesting part of the story, and I think we still don't know exactly how to interpret it in terms of photosynthesis. Particularly, is this evidence? I mean, is it evidence for oxygenic photosynthesis? I think that's really the issue. That is oxygenic photosynthesis in the Archean very early. Okay, what else is on this slide? All right, they're just showing roughly the time of arrival of various processes and organisms. So again, this late rise in oxygen, which we're not going to get into here, but is quite interesting really in the Neoproterozoic, is apparently associated with land-based eukaryotes. These over here, and vascular plants, perhaps a bit later, but in fact certainly later. But it's a worthy topic, and I just sort of completely beyond my knowledge. So I'm just not going to worry about it. Okay, so moving on. Okay, probably the most quantitative record we have for the occurrence of oxygen in the atmosphere is this mass-independent fractionation of sulfur isotopes. So this quantity here, this Cap33S, I guess most of you are familiar with this by now, for the most part. I don't want to get into a bunch of detail about this. I work in depth on this topic, and it'd just be a bit of an aside for what we've got at the moment. But this is a measure, this cap delta 33S is a measure of excess 33 sulfur in the given sample that you're looking at. And so in most samples, if you look in the first, from essentially present day up to 2.4 or so billion years, sedimentary rocks and virtually everything, organisms, just everything we look at in sulfur isotopes has no unusual excess of 33 sulfur. It has the expected amount based on isotopic equilibrium arguments. Those arguments are applied to rocks normally, but you can apply them to other things as well. But when you go back beyond 2.4 billion, you suddenly see this quite noticeable rise in excess 33S, meaning positive F33 values, and also in negative 33S values. These are mostly in barytes here, there's mostly in pyrites, and there's a lot of speculation about what exactly is causing this. The interpretation that was offered initially, and that is still essentially the interpretation, is that this reflects SO2 photolysis in the atmosphere. And the difference was that here, where you're seeing this signature, you would photolize, you can photolize SO2 in the atmosphere, a volcanic SO2 or whatever SO2 at any time, but prior to an oxygenated atmosphere, when you photolize that SO2, you would produce essentially two reservoirs of sulfur, one of them being elemental sulfur and the other sulfate, once you oxidize that SO2. And because all of the sulfur was not oxidized into one reservoir, it is possible to produce an isotope effect in the photolysis process. This is the part that I work on in some detail, and preserve it. Preserve it because you actually have two reservoirs. You have the elemental sulfur and the sulfate, and they don't do the same thing when they get into the ocean sedimentary environment. Now, in the present time, and since we've had an oxygenated atmosphere, all of the sulfur is, regardless, you can photolize SO2, you photolize H2S, CS2, anything that you put in the atmosphere, sulfur-bearing, that is, and you photolize that or photoprocess it in any way, it all ends up oxidized and converted into sulfuric acid. You have one reservoir. Anything that you've done photochemically to impart a sulfur fractionation to that, in any of those steps, is simply erased because everything is added back together and stuck in one compound, and that is essentially sulfuric acid, sulfate. So we lose that signature all through here, but we still have it here. And so this is used as just a real hard marker for the rise of O2 in the atmosphere. And it certainly is a very sensible interpretation. There are details in terms of working out exactly whether you get the expected signatures from SO2 photolysis and all that one expects, that one sees in the rocks. Do you get that when you actually photolize SO2 in the lab? That is an in-depth and heavily worked on topic at the moment, by myself included and some of my spectroscopy colleagues. So, but right now, the interpretation, and I think it's quite reasonable, is that this right here marks, somewhere very close in here, about 2.5 billion years, marks the transition from very low O2 levels, less than kind of ppm levels, to something above that. So, it might only be 100 ppm or something of that sort, but it was high enough that it it affected the atmospheric processing of SO2 and turned everything into sulfate. Okay, so there's, of course, a relatively spotty record. There are details in here, different variations in the magnitude of the signature, all stuff that can be explained in various ways that I'm not going to get into now. But it's a very powerful record and has had a profound impact on how we interpret oxygen, the arrival of oxygen in the atmosphere. Okay, so getting on to then photosynthesis. Those are really, I think I left out the iron isotope stuff, but those are the principal geological constraints, geochemical constraints we have on photosynthesis. Left out the oxygen, the carbon isotopes for anoxygenic species as well, but Okay, so then what about photosynthesis? Now this is going to be just a very simple overview. There are some experts on the phone here who I invite to jump in and correct something if I don't say it in enough detail, or if I say something incorrectly. So this is, what I'm showing here is one of the simplest light gathering systems we have. It's not photosynthesis. This is a rhodopsin system. This is a light-driven energy transduction system, if you will, and so what I'm showing here is a membrane layer, and light indicated by these yellow bolts, lightning bolts, and the rhodopsins take in actually the rhodopsin protein is down here, this little picture. So it's a heptamer of alpha helices connected by loops. And inside, in the middle of those alpha helices, is a retinol pigment. So a carotenoid is sitting in there, capable of absorbing visible light. And this, move that down to remember to move the right pointer. Okay, there's the retinol there. It absorbs a photon, visible photon, at an appropriate range of wavelengths, and undergoes isomerization, which allows for proton transduction through this channel, through the rhodopsin. And so you actually get light-driven movement of protons into the cell, and ATPase, which is also an integral membrane-bound structure, is able to take those protons and use it to drive ATP synthesis. Okay, so it's really just a beautiful system. It's so simple, taking photo energy, light energy, and converting it directly into chemical energy used by the cell. As we all know, ATP is just of such fundamental importance in every single organism that we know of. It's almost hard to overstate it. But so this really is a marvelously simple and effective system for ion movement, for ion transduction. And it's used for chloride ions, used for protons, as I just mentioned, and it's fairly prevalent, but it's not as prevalent as one might expect. It's primarily in archaea, I believe. Again, someone in the audience here can correct me if I'm wrong about that.
It's also widespread in certain proteobacteria.
These are the marine proteobacteria, I think.
Yeah.
Yeah. So I have to say, as someone who hasn't thought as much about these topics as some of you guys have, I look at this and I just think, why isn't this used in some fashion more? I mean, it's just so elegant and so, I like the simplicity of it. It just kind of surprises me, but there are reasons, I guess. Anyway, this is, I'm not going to spend too much time talking about this, but one can't help but imagine that something like this would have developed certainly well before oxygenic photosynthesis with its double photosystems and all. This is just so much simpler. But of course, this does not get you reducing potential, reducing equivalence. So you're not converting CO2 into glucose from this process. This is just to generate ATP. Okay, so that's
you can use that ATP to drive a reverse electron flow to do reduction, right. And so that's how the purple photosynthetic bacteria do it. And so, in principle, this could provide the energy for carbon fixation. Right. In practice, I don't know of any organisms that are real photoautotrophic using rhodopsin-based photosynthesis. It just doesn't seem to give you enough oomph to really power your whole metabolism.
Okay. Is that partly because it's not such a great collector of photons, or is there some better reason for that?
Well, it only uses a fairly narrow region of the solar spectrum, so you don't have a lot of solar flux that you can harvest using these pigments, although you could imagine that evolution could have broadened it out, as it has in a lot of visual systems.
Exactly.
I don't have a good explanation for why. I agree with you. It's a beautiful, elegant, simple system, and it's surprising that you look out and the world isn't purple. But it is the case that you look out in the world as green, and so while
yes,
this
it is
obviously an important and possibly very early system, it hasn't become the dominant mode in the biosphere today, obviously.
Right. Okay. All right. Then the systems that are in fact dominant are just very generically indicated here. Again, taking figures just straight out of Holman, Marriott, and Blankenship's paper, 2011. Nice review paper on evolution, photosynthesis. What's shown here in this one labeled B is again photons coming into what's called a Q-type reaction center, the quinone type reaction center, and this is a very simple system in that it's utilizing cyclic electron flow. Okay, so my pointer is here. Okay, there we go. Cyclic electron flow here. And it's more or less, I would say, the simplest of the true photosynthetic systems, not counting rhodopsin as a true photosynthetic system. And although similar, again, in that it's really then you're just using this energy, this proton gradient generated to produce ATP. Okay, and I think, remind me, this is a fair summary of the purple bacteria, or is that not correct?
Yeah, that's true.
Okay. All right.
Yeah, that's meant to be the purple bacteria.
Okay.
The top B.
Right, exactly. Okay, and then the lower one is, now this is same situation, photons coming in, reaction center here, now using iron-sulfur cluster. It's iron-sulfur type reaction center, so as opposed to the Q-type, the quinone type reaction center here, so different electron carrier. And
this is meant to be the green sulfur bacteria.
This is green sulfur, yes, and the
Yeah.
point here is this is just creating reducing equivalents to be used in carbon and nitrogen fixation. So this really is, I guess, what we would call, it's closer to photosystem 1 in some ways, right?
That's right.
But you have to realize that those guys are also doing a cycle as well.
So there is a cycle. There is you have to return electrons, right?
Well, I mean, you need to pump protons to get to make ATP, so that's going to be going on as well.
I see what you're saying, yeah.
In addition to the electrons that are going out of the cycle to drive carbon and nitrogen fixation.
Right. Like Kevin says, they're going to have both. But interestingly, I don't think we really understand the kind of partitioning between those two modes of electron flow very well in those kinds of organisms.
Or at all.
The least understood in that respect.
All right. The least understood in which you mean this, where you have both flow to create reducing equivalents and the return flow? I'm not quite sure.
The cyclic versus the non-cyclic flow and how that works in these organisms.
Okay.
There's a huge gap in our understanding about that.
Gotcha. Okay. All right. Okay, well, this is just a generic view of reaction centers. I shouldn't say generic, a schematic view of reaction centers. Again, different organisms here. So the purple bacteria, cyanobacteria, this is photosystem 2 of the cyanobacteria, photosystem 1 of the cyanobacteria, and the green sulfur bacteria, and the heliobacteria. And the idea here is, I'm not going to get into this in any depth, but just to show what the reaction center actually looks like. Each of these circles you see are an alpha helix, so it's a membrane, integral membrane spanning protein. And there are each of these reaction centers. Okay, this is what I'm referring to. This is the alpha helix here, each of these circles. And each of these reaction centers is composed of a dimer of these transmembrane proteins, 5 in each dimer in this case. And some of the dimers have some differences. That's what's indicated here by the red versus blue, so they refer to this as a heterodimer. And some are very similar and referred to as a homodimer. And I believe most of the differences in the hetero versus homodimer are in quinone transfer. I understand that correctly, which I'm sure I have a rather simplistic understanding of it. That some antenna structures are here as well, right in the reaction center. And of course, there are more peripheral, more distant light harvesting proteins, pigments in the membrane also. Okay. So the idea here is just to illustrate what these reaction centers look like. There are many things in here as well. I mean, this is where this is really where the action is. This is where the charge separation occurs, and there are quinones and chlorophylls. This is the quinone type reaction center here, the Q type. Various electron transfer transport elements, and many. I cannot do justice to this, but and then when you move over to the more photosystem 1 type, or the type 1 reaction centers, you see iron sulfur clusters involved as well. And again, lots of stuff going on here. There's been a huge amount of work on this over the years, and the evolution of this, I would have to ask Bob or Kevin to speak to the evolution of these kinds of systems if they wish to, because it's simply beyond my knowledge. But it's quite clear that there's been a lot of lateral gene transfer in these systems. They're related in many ways, and that is, I think, fairly straightforwardly.
Yeah, I could just comment on a couple of things, and then I'm sure Kevin can too. One of the key evolutionary developments is the issue of the heterodimerization of these systems. So where you have the red and the blue there on the left, that represents a heterodimer protein, which is a protein that has resulted from a gene duplication and subsequent divergence to create two similar but not identical copies of the core reaction center protein. And you see that in the purples and in the cyano PS1 and PS2, but in the green sulfur bacteria, heliobacteria, and acetobacteria, you have what's called a homodimer, and that's where you have a single gene that has not duplicated, and so you have two copies of a single gene product that associate to form the reaction center complex. And that presumably represents an earlier form of evolution prior to gene duplication events.
So functionally, what is the difference between the homo and heterodimer then?
Well, in the Q types, you have electron flow that goes down just one of those two branches down at the bottom left there. In the PS1, and Kevin can speak to this better than I, you do have electron flow that goes down both pathways. And so the functional importance of the heterodimer in PS1 is not very clear, at least not to me. And in the ones that are homodimers, almost certainly you have equal pathways such that either pathway can be utilized, but we don't have any real good structural data on those.
Okay. Yeah. I would just add. I mean, I agree with everything Bob just said. For the type 2 reaction centers, the quinone type, the Q type, clearly the heterodimerization there is important for its function because they only use one branch for electron transfer, and they stabilize the semiquinone on the other side and then eventually double reduce it. So that's part of their mechanism. And I imagine that heterodimerization happened early on for those guys. In fact, it probably happened independently in the lineage leading to PS2 and the one leading to the purples. For PS1, though, it's a different case. Actually, even though they have heterodimers, like Bob said, we've shown for a while now that they use both sides. And actually, most of the differences seem to be not in the middle where the electron transfer is occurring, but more on the outside in order to make an asymmetric binding site to optimize binding of electron donor acceptors outside the membrane. And actually, some work that we've done and John Goldbeck has done kind of agrees that they may have done this in response to the presence of oxygen. So that is that PS1 may have evolved from a homodimeric ancestor in response to the presence of oxygen. That's an idea, but I think it fits the data pretty well.
That's an interesting. That would be good to test. Okay, so I'm not going to. We're actually taking quite a bit of time here. I'm not going to go through the redox energies of these systems. I think if somebody wants to see that in some detail, just ask. It's well known for the different systems. So let me literally just skip this for now. This is what we were just discussing. These are individual. Sorry, I'm finding the pointer. These are individual alpha helices here, just showing the process, possible evolutionary processes of fusion and duplication, divergence, all the different things that can go on genomically to create different types of reaction centers. Okay, and so let's push on to what the questions are, because we have this long list of questions to go through. And so I just copied the questions, and I'm just going to mention them. I'll just read them, and with small comments added. So, looking at the geochemical fossil evidence, what are the big issues related to photosynthesis? One is the carbon isotope signatures in these graphitic inclusions in old rocks, the oldest rocks, 3.8 billion-year-old rocks, Keelia, they're about minus 20 to 30 per mil relative to carbonates, as we talked about initially, which is it's consistent with thermal processing of photosynthetically derived organics. But can this be an abiotic process also? And this has been looked at somewhat by some folks. But I think it's something that needs to be addressed in even more detail. And Jack Hills, if we go to the Jack Hills zircons, these are the oldest rocks, if you will, that we have. They're really minerals, of course. But these Jack Hills in Western Australia, these zircons are 4.3 to 4 billion years old, based on uranium-lead ages. And they apparently, I did not know this, but they contain evidence. They contain some, I should say, do they contain contemporaneous organic graphitic matter? They do contain something that is, they do contain some carbon, let's put it that way. Whether it's truly contemporaneous or not, I don't know. If it is, and it certainly would be good if it is, then someone's going to measure the carbon isotopes at some point. I don't think that's been done yet. Certainly should be done. And if it comes out in this minus 20 to minus 30 range, is this, again, evidence for life and photosynthesis in this very early, now we're talking about a very early window, this 4.3 to 4.0. I mean, this is really the only handle we have at the present time on carbon isotopes from this pre-3.8 period. So it's very important to at least try to do these measurements, and then we can argue about what it means, if it means anything. But it's certainly there's no other way that I'm aware of to get at that time period on Earth, apart from finding lunar crust blasted off in an impact on the moon from that age. But good luck finding that. I mean, it's probably there, but it'd be hard to find. Okay, another question. Are the apex chert microfossils really cyanobacteria? How can we differentiate these? We can't simply assert that they are because they happen to have some morphological similarity. There has to be something a little better than that, and I don't know what that is, but that's something that needs to be also dug into in a bit more detail. Bill Schopf may say that that's a solved issue. I don't know, but it doesn't seem to me that it is. Number 4 here, were banded iron formations primarily a product of anoxogenic photosynthesis using ferrous iron? Okay, this is something being actively worked on via iron isotopes and perhaps other methods as well. So I think that there will be progress on that, and has been to some extent already. And sulfur mass-independent fractionation argues for oxygenic photosynthesis by at least 2.4 billion years. But how much earlier could it have been? And maybe it's more accurate to say this is about 2.5, but you know what I'm talking about, that break in the curve. where you go from zero mass-independent fractionation to suddenly quite dramatic mass-independent signatures as you go back in time. But how much earlier could this have been? There is some evidence from transition metal abundances, manganese in particular, for small amounts, low levels of oxygen at various times, going back to about 2.5, maybe 2.7. And it's possible that oxygenic photosynthesis was actively going on for quite some time, but the oxygen was being titrated out by the ferrous iron in the ocean, and did not have a chance to appear until a good chunk of that ferrous iron was removed, and in fact much of it is removed by not all of it, but much of it is removed by about 2.4, 2.5. Or was it simply the case that there was no oxygenic photosynthesis at that time prior to 2.5, 2.6? Again, we don't know. And this something that I originally, once I think many of us were very hopeful for these organic geochemical markers of the hopanoids and things of that sort, representative of various membrane-bound sterols and other fairly good-sized stable molecules, that these would be useful as markers of anoxygenic oxygenic photosynthesis going back in time into early Proterozoic, into the Archean, and that was indeed the case for a while, or at least we thought so, but there has been an issue of contamination regarding the interpretation of all of these organic, I shouldn't say all, but many of these organic geochemical markers. And so the field, that whole field is in a bit of a state of flux at the moment, and I don't know exactly where that's going to go, but it certainly is an important component of all of this. Okay, so what about prebiotic chemistry? Now, we're talking about origin and evolution of photosynthesis. Could you do it in a prebiotic sense at all? Certainly it looks so complicated that the answer is almost certainly no, but we can ask some simple questions, things like, number one here, were prebiotic chromophores present in the surface of the Earth, in photoxomes or wherever? And that's certainly a plausible thing. Some carotenoids are not that complicated. They could be present in various forms. Porphyrins perhaps less so, but even they are not wildly complex. Certainly more complicated than methane or something like that, but it's not implausible to imagine having some of those compounds present prebiotically. Were simple electron chains possible? So imagine now that you have membranes, you have cells, but you may still not have living systems. Would it be possible that you could get some chromophores in those membranes? You could perhaps do some excitation via photons and transport. This is all very well-tuned stuff now when we look at it, all of the excitation transfer and all of that goes on in pigment systems. So to imagine that could just pop up prebiotically is perhaps pushing it, but simpler forms of that are possible. Quinones are not that complicated. Iron-sulfur clusters, we know, can form in plausibly prebiotic scenarios. So it is a sensible thing to consider, I would argue. Did delayed heavy bombardment delay the onset of photosynthesis? The delayed heavy bombardment was, just to remind everybody, if I imagine most know, was really from about 4.1 to say 3.8 billion years. And in and of itself, it would not have created a molten Earth or anything like that. It would just have created brief spikes of high temperatures, and of course melting if you're in the area nearby where any asteroidal-sized object is impacting. But if you do, the late heavy bombardment does consider the possibility of very large impactors, say on the order of several hundred kilometers, that could have vaporized the oceans. If you vaporize the oceans, you may have to reset some of the biological processing, and assuming that it was present at that time of the impact. So it is possible, especially given that photosynthesizers are right there at the surface and see everything going on near the surface, that they could have felt the effects of these kind of good-sized impactors. I don't know how we're going to address that because of the early time period, except by those Jack Hill's zircon sort of measurements looking for carbon isotopes. Let's see. Other prebiotic questions were integral membrane proteins, that is alpha helices, produced in an RNA-DNA protein world. Certainly they shouldn't be made in an RNA world, but they could be a product of an RNA-DNA protein world. And even one that is, depending on how you define life, it might still be a prebiotic world, but one can debate that. But they're not, alpha helices are not that complicated, and it's a certain set, prefers a certain set of residues, but they're fairly straightforward. And they can, in some cases, pop into membranes. Okay, so now getting into more of the evolution questions, and again, I will, my colleagues can expand upon any of these that they feel they would like to. So, origin of anoxygenic photosynthesis, it's presumably from earlier heterotrophic or chemoautotrophic organisms. We're not claiming that photosynthesizers were the origin of life. That would be a bit pushing it, but indeed they could come from heterotrophs and other things. So, questions related to that then, what was the ancestral reaction center, and what was its origin? The reaction centers, as we just discussed, are complicated. They're not crazy complicated, but they're pretty complicated, and clearly they are well-evolved structures. Can one make a reaction center with one or two membrane-spanning proteins? And can you begin to approach that? Of course, you have to bind pigments and all of that, but that's the sort of question that one wants to ask. Was the ancestral reaction center a simple homodynamic photosystem? Okay, I think Bob already answered that, saying it's pretty likely that these homodimeric reaction centers are thought to be earlier. Did iron 2+ sulfides or other easily oxidizable compounds act as primary donors in the reaction center? What we're looking for is evidence of that. It seems like a logical assumption. This I stole right out of one of Bob's papers, just a marvelous question for its directness and simplicity. Was the last common ancestor photosynthetic? Wonderful question. I don't know. It certainly seems sensible, but I don't know.
I think the answer is no. I think the answer.
You think the answer is no?
Yeah.
The last common ancestor.
Luca.
Yep, go ahead.
The reason I say that is that if you look at the distribution of at least chlorophyll-based photosynthesis in the tree, you find it all in the bacterial domain, and then, of course, the endosymbionts in the eukaryotes. You don't find a single case of archaeal chlorophyll-based photosynthesis. So to me, that suggests that the chlorophyll-based photosynthesis was invented within the bacterial domain and then spread to the eukaryotes.
Okay.
But you could get out of that if you really wanted to. But that's what that distribution in the tree says to me.
Right. All right. Are rhodopsins more ancient than photosynthetic reaction centers? Again, the basis for the question is because rhodopsins and their capability of generating proton gradients, light-driven proton gradients, are so much simpler than photosynthetic reaction centers. One has to ask, are they in fact more ancestral? Are they more? Did they appear earlier in an evolutionary sense? And I don't know that we have any evidence one way or the other for it, but it sort of begs the simplicity of the rhodopsin begs the question. Did reaction center complexes originate from a concatenation of one helix proteins with pigment binding sites? That's really one of the figures from Wim Vermaas' paper. It certainly sounds plausible. I could not really address this in detail, but if anyone, Bob or Kevin, want to add to that, expand upon that, feel free to do so. Sounds like a sensible thing. This is just a basic question about environment, really. Did oceanic photozones before 2.4, in other words, before we had an oxygenated atmosphere, provide enough UV shielding to allow proteins and nucleic acids to avoid UV damage while maintaining a high enough visible flux. And maybe you can get around this with some pigment, UV absorbing pigments that can protect the organism. But if you did not have those, then you would have to be deep enough in the ocean, in the water column, to avoid significant UV damage. All right. Origin of oxygenic photosynthesis. We're almost done. Yep, we're almost done here. From anoxygenic photosynthesis. Okay, oxygenic from anoxygenic. Again, seems like a reasonable assumption that oxygenic came after anoxygenic. Just a few quick questions here. What was the selective pressure for forming two types of reaction centers? And on the surface of it, it seems like it's simply that if you want to use water as a source of electrons, you're going to, you're covering a fair span of energy there, so redox potential. So you really do need some way to do that. But maybe that's just a completely naive point of view, and there's some more subtle reason for it. Did oxygenic photosynthesis arise from a reaction center with a highly oxidizing primary donor? In other words, could you get away with using just one reaction center? I have no idea. The answer to that is unlikely. What driving forces led to heterodynamic reaction centers? And this gets back to a little bit of our earlier discussion. And how did that should be a the, not a he. How did the oxygen evolving complex evolve? Them just asking this question, and I do not know anything about the oxygen evolving complex, so if anyone wants to comment on that, please do. And finally, again, this is a very interesting question. I believe I got it from one of Bob's papers. Are there undiscovered types of photosynthesis in photosynthetic organisms present on Earth today? Almost certainly there were some present once upon a time, and we have long since lost that information. But are there any around today? I believe that was the point of the question that Bob was posing in that paper. But it's certainly an interesting possibility. Okay, and I believe that is it. I think we've gone for a full hour, which is a little longer than these usually go for. So.
Well,
hey, Jim, I'm going to add about that last point about oxygen evolving complex.
Yeah.
Some work from Woody Fisher at Caltech is you can interpret it in light of the idea that you might have used manganese as an electron donor, like some organisms still use iron 2 as an electron donor now.
Yes.
And that may have occurred prior to the rise in oxygen. So you might have had reaction centers in which they were using manganese as an electron, just as an electron donor, and from that, if it became oxidizing enough, you could have evolved an oxygen evolving complex as the oxidized manganese bound to the reaction center. So Jim Allen here at ASU is doing some work on mutating purple bacterial type 2 reaction centers to bind manganese, and you can actually see them oxidizing manganese.
Okay.
They're not forming an oxygen evolving complex, of course, but that's not so far-fetched.
We actually tried to determine if there were some modern cyanobacteria in a natural environment that was oxidizing manganese in a hot spring, but we didn't really find any evidence of that, at least in situ.
Is this Lindsay? Or?
Oh, no, sorry, this is Nikki Peranto.
Okay, yeah, please identify.
Yeah, sorry, I work with Bev Pearson on that one.
Okay. And could you say that one more time, please?
Sorry. We were working at a hot spring in Yellowstone that has very high levels of manganese as well as Fe2. And so we were asking, do these cyanobacteria use Fe2 directly? And also, do they use manganese? And while we found evidence that they did use Fe2, we didn't find any evidence of manganese oxidation, at least in situ in those natural mats.
Did you look at any anoxygenics, which might have had more of a pressure for electron donors?
Yeah, there's chloroflexus and rosiflexus in there as well, but they're not doing anything. They're growing photoheterotrophically.
Usually they are, yeah. Mm-hmm. Okay, thanks.
Mm-hmm.
Okay, and this is Nikki, right?
Yeah.
Yes, okay, I see you. I just now noticed your comment about the hydroxyprobionate pathway for chloroflexus, carbon isotopes, about minus 15 per mil. So yeah, that is about 10 per mil less than
they're pretty
the
heavy.
Rubisco pathways. All right. Okay. Any other questions? Shall we? And again, I apologize for not having. I have two cameras sitting here attached to my computer, and I could not get either one working. So we just had to do this by voice. Okay. If no other questions, I think we're all set.
All right, James. Thank you very much.
Okay. Thank you very much
for wrapping up our series here. Lindsay is going to be sending out an email shortly on next steps because we know that you'd all miss us if we didn't. So expect to hear from us shortly, and then we'll be outlining the next steps in the process after that. So thank you very much, everyone. Cheerio now. Bye-bye.
Okay. Thanks, Andy.